Rachel May Chen. I'm a professor at UC Riverside. We were one of the first few labs to have discovered the existence of microRNAs from plants. MicroRNAs are tiny RNAs that are powerful regulatory molecules in plants. And they were like the dark matter in biology. Their existence in plants and animals was not known until 2001. And we didn't know anything about how they work, how they're made, how they're degraded, etc. And in the past 15 to 16 years, my lab has been working on how these molecules work, how they're made, how they're degraded, how they exert their regulatory functions. Potentially, this would set the foundation for using these molecules to better agriculture and medicine. As you see, we have lots of plants that we grow and study. We study how the molecules of these plants actually work, like the DNA, RNA, and proteins in these cells, in the cells of these plants. And we actually learn a lot from these plants, even about ourselves, because these molecules actually are very similar in their function in both animals and plants in our lives, but we probably don't all the time, right? But we probably don't pay much attention to them, right? But plants are so important to us. Just take a look at the food on your plate, right? You know, we can't survive without plants. And these days, because of the increasing world population and because of climate change affecting agricultural productivity, you know, there's more urgency to study plant biology. We need to understand how plants work, you know, how they respond to heat, to cold, to stresses, how they decide when to flower, so that we can better agriculture. And in fact, you know, we also learn a lot about animals by studying plants, because a lot of the processes that we and others study are common in plants and animals. And you know that it's hard to use humans as organisms to study, right? But we can use plants to study these basic molecules, and that the knowledge from plants would also inform us about ourselves. Global warming, you know, is going to affect plant productivity, because, you know, plants that used to grow at this temperature probably cannot adapt very quickly to this changing temperature. So we need to understand how plants actually respond to temperature. And hopefully we could either breed or engineer plants so that they could respond to high temperature better. Mm-hmm. So hopefully by studying these plants, we can understand how plants in general adapt to hot temperature. And we could try to engineer plants to help them better adapt. By the way, so this little plant is called Arabidopsis. It's a plant in the mustard family, Brassicaceae family. For example, broccoli, cauliflower, bok choy that we have as vegetables, they belong to the same family. But this plant is being used by many plant biologists to study plant biology. And that's because it's very easy to work with them. They're small. We could we could manipulate them any way we want. And the nice thing is that there's a common law underlying plant biology. You see plants that are as high as a sequoia tree or as tiny as Arabidopsis. But common laws underlie their cell biology, their molecular biology. So by studying this little plant, we can actually know a lot about these common laws that govern plant biology. Here we have only two plants.
Oh.
These are all Arabidopsis plants, and these plants here, these are tobacco. So we use them to do our experiments. So basically, you know, plants undergo different stages of development, right? So this would be a young Arabidopsis plant, and this is a few weeks old, plants a few weeks old, and these are plants that have flowered. And then these are the seed pods that are made. And from the seed pod, we get a lot of seeds, we plant the seeds, and they become something like that later. So that's the life cycle that you're looking at.
Oh,
yeah. So these are pretty much all Arabidopsis plants. And you cannot imagine how much we have learned from this tiny little plant. Yeah.
I know. I guess every plant has a story of its own. By studying them, it kind of reflect our human being, how the evolution evolved, right?
Exactly, and also how they develop, right? Like from a seed to a vegetative plant to a flowering plant, and how they make seeds and make flower, how they make the next generation, basically the seeds, right? So we learn a lot about plant biology from this tiny plant. The main thing is that there's, being either Arabidopsis or tobacco, these molecular events that govern plant biology are very similar. Mm-hmm. So by studying this plant, we know a lot about that plant. And by studying this plant, we even know a lot about what's going on inside us.
Right.
Yeah.
So how long do you usually spend a day? How many hours you spend a day here in the laboratory?
Oh, in the laboratory, for example, my postdocs and graduate students, they, as long as they are not, the students, as long as they're not in classes, then they spend their time in the lab. Me, I teach and I also do research. So I would say maybe 50% of my time is on research.
I see.
Yeah,
I read there's a lot of articles you wrote about research about your plants. I guess what is the most proud moment, I guess, for you for all this past?
I guess the most proud moment for me was the identification of these microRNAs. We don't see them, they are hidden. Yeah, they're hidden in those plants. They're tiny little RNAs,
right.
And they were like the dark matter in biology. We didn't know they existed until 2001. And the most proud moment for me was the time when we realized that we had discovered microRNAs in plants for the first time. And starting from then, we were trying to understand these tiny, mysterious molecules. Yeah, these are tiny Arabidopsis plants grown on plates. We have medium, a thin layer of medium, and then we put seeds on there. They germinate. Now these are like 12-day-old seedlings. And now Lorena is going to transfer these seedlings into soil.
Oh, okay.
Yeah.
So let's take a look.
Yeah, take a look at these plates. And she's transferring them into soil. And then later on, they're going to grow and then flower and make seeds. And we use them in our studies.
So how long does it take for it to grow into this?
To this big, about 2 weeks.
2 weeks.
Yeah.
And then you transfer over to
soil.
And then the soil, is that where you guys put into the...
Exactly, that growth room.
Can you talk about a little what are they doing?
You see, Cui, a postdoc, she's now putting these tiny plants into agar. We then later section them. We cut very thin slices of them, and then we put them in under microscope to observe the cells. And we have actually fluorescent proteins expressed in them, so that we could see where, in which cells these fluorescent proteins are present. After she puts these tiny plants under the microscope, we will be able to see the fluorescence, the green fluorescence. This represents the expression of a particular gene only in those cells. Yeah, so what you're looking at is the top-down view of the growing tip of the plant. So these are basically leaves, and you see that this fluorescence is only in a few cells in the plant. That's how we trace the expression of certain genes. Fluorescence from this GFP, it's actually a protein from jellyfish. Jellyfish, yeah, but we use that to mark gene expression in plants.
So by studying this, what you try to accomplish?
We try to know what genes are acting in what cells. Yeah, for example, we were talking about global warming. So when you have hot temperature, what genes become expressed? So these are the genes that may be responding to hot temperature, right? So we're trying to study the genes that are underlying certain biology in these plants. Yeah. As I receive this award, I would really like to look back to remember and thank the people who impacted my life. The first significant event in my life was the opportunity for me to attend Peking University to study biology. For this, I would like to thank my parents and my grandma for providing me a happy childhood and for believing in me, having the belief that girls are no less worthy than boys. The second most important event in my life, I would say, was the opportunity to pursue PhD at Cornell University. And I entered Cornell University through the CUSPIA program, the China-US Examination Admissions Program. This program was run for a total of 9 years, and it enabled about 450 students from China to pursue their PhD in US institutions. And I would like to thank the organizers of this program, the late Professor Ray Wu and Professor Xiaocheng Gu. Ray Wu from Cornell University and Xiaocheng Gu from Peking University. To the young generation of Chinese American scholars, I would say that find your interest, pursue your interest, put in the efforts, and do not give up. And you are the future.